Hi, my name is Keely Butters, and I am an undergraduate senior at Towson University. My project is called Mutations in the Furin cleavage site to improve cytotoxicity in anti-mesothelin recombinant immunotoxins based on Pseudomonasexotoxin A. So to start off, a recombinant immunotoxin is comprised of the variable fragment of an antibody and a toxin. The recombinant immunotoxin I am using for my project is made of SS1-LR, which is an anti-mesothelin antibody, and the AB domain of Pseudomonasexotoxin A, which is derived from the opportunistic pathogen Pseudomonas aeruginosa. Recombinant immunotoxins can be used for targeted cell therapy, and recombinant immunotoxins based on Pseudomonasexotoxin A, or PE, are currently being used in clinical trials to target cancer cells. Next, we will look at the intoxication pathway of this recombinant immunotoxin. Once the antibody has targeted the cell, the recombinant immunotoxin will bind to a low-density lipoprotein receptor-related protein that is found on the surface of the cell. Once bound, PE will enter the cell through cell-mediated endocytosis and will travel through the cell using a retrograde trafficking pattern to reach the cytosol. When PE reaches the endosome, it encounters the protease Furin. Furin cleaves the PE recombinant immunotoxin, which activates the toxin. Furin will cleave the recombinant immunotoxin at the region connecting the antibody variable region and the toxin. The antibody will be sent off for degradation, and the toxin continues on through the cell, halting protein synthesis. This leads to cell death of the targeted cell. Unfortunately, furin is not quite efficient at cleaving the recombinant immunotoxin, which is a problem that my project focuses on. The purpose of my project is to create mutations at the furin cleavage site in anti-mesothelin PE-based recombinant immunotoxins in order to increase cleavage efficiency and improve cytotoxicity. I hypothesized that the mutations N-terminal to the furin cleavage site will increase both cytotoxicity and cleavage efficiency, while mutation C-terminal to the furin cleavage site will increase cleavage efficiency without significant improvements to cytotoxicity. In the chart here, the P mutations are the mutations that are N-terminal to the furin cleavage site, while the P' mutations are the mutations that are C-terminal to the furin cleavage site. The furin mutations are simply the P and the P' mutations combined, and the wild-type sequence is, of course, the sequence that has no mutations in it. The mutations in the chart are based off previous results in a related study. To achieve this goal, we have to create the recombinant immunotoxins with the selected mutations. We start off with a DNA insert that contains the selected mutations at the furin site. We then express this in a plasmid and transform the plasmid into NAB5-alpha competent E. coli cells. We will grow the cells overnight and extract the DNA. Once we have extracted the DNA, we will send the DNA construct off for sequencing in order to confirm that we have the appropriate mutations. We will do this for all three of our DNA constructs. After we have confirmed our mutations using DNA sequencing, we will then purify our proteins. We do this by transforming our proteins into BL21 competent E. coli cells and then expressing our proteins. After the proteins have been expressed, we'll refold the protein and purify it using size exclusion chromatography and anion exchange chromatography. Once we have our purified proteins, we will then look at how the mutations in the furin cleavage site affect cytotoxicity. We do this by creating cytotoxicity assays that contain L55 cells. These are lung adenocarcinoma cells, and since mesothelin is a protein that is found on the lung, these cancer cells would work well with the anti-mesothelin antibody SS1-LR. Once we perform the cytotoxicity assays, we will read them using a microplate reader at 450 nanometers wavelength. We ran 4 cytotoxicity assays with L55 cells for each of our 3 recombinant immunotoxins and the wild-type SS1-LR-GGS. We determined the EC50 values of each of the recombinant immunotoxins, took the log of the EC50 values, and plotted them on the graph here. You can see that the SS1 LR Furin mutations and the SS1 LRP mutations, which are the N-terminal mutations, had decreased cytotoxicity compared to the wild-type SS1 LRGGS, and the SS1 LRP prime mutations, which is the C-terminal mutations, performed similar to the wild-type. We ran a paired t-test of the EC50 values of each recombinant immunotoxin and only found significance between the wild-type SS1-LRGGS and SS1-LRP'. We determined significance by looking at the p-value, which was 0.0244. This is less than 0.05, which would indicate significance. After analyzing the results of the cytotoxicity assays, we concluded that the mutations N-terminal to the cleavage site, which was SS1-LRP, decreased cytotoxicity relative to the wild-type SS1-LRGGS, while the mutation C-terminal to the cleavage site, SS1-LRP', had a minimal effect on cytotoxicity. The t-test suggested that there was a slight increase in SS1-LRP' activity, which could indicate a slight increase in cytotoxicity compared to the wild type. These conclusions indicate that my hypothesis was incorrect. The next step in the project would be to recreate these mutations in a recombinant immunotoxin using a different antibody, HA22LR, which is an anti-CD22 antibody. I have already started creating these recombinant immunotoxins with the HA22LR antibody, but I have not yet tested cytotoxicity. I would like to thank the biology department at Towson University, as well as Dr. Weldon for his mentorship and guidance, and Jillian Baker for all her help and guidance. I would also like to thank Danielle Reefer and all other members of Dr. Weldon's lab. And this is my bibliography page. Thank you for listening to my presentation, and I hope you have a great day.